So now we're going to kind of shift gears to how we can control microbes. We had to understand how transcription and translation and those processes occur in microbes so that we know how we can actually disrupt them and stop those from occurring, right? So with the recent pandemic of COVID-19, we're currently very aware of the need to control our exposure to harmful microbes. And so we've got a little bit more hypervigilance these days. This is and has always been, however, an ongoing concern. This is a very important slide. Know this slide. So the methods of microbial control used outside of the body are designed to give us these four possible outcomes: sterilization, disinfection, decontamination, also called sanitization, antisepsis, otherwise known as degermation. Okay? Understanding the difference in these is important, especially in the healthcare setting. We use these terms often interchangeably in our everyday life, but they do have different meanings, and we need to understand these. They have different applications in microbial control. Sterile should refer always to only materials that have been through the process of sterilization. That means all viable microorganisms, including viruses, are destroyed. They have undergone heat, a strong chemical agent that is strong enough to destroy an endospore. This is commonly done on surgical instruments, syringes, or packaged items. This is not done on human tissues. Disinfection is the process that destroys pathogens in a vegetative state. Okay, this is, let's see, all viable microorganisms, vegetative state, okay, but not endospores, all right? Not endospores. So this is also used on, it is also not used on human tissues, but this is used for things like utensils, thermometers, those types of things. And this is often done with bleach solutions, iodine, or by boiling, okay? Decontamination or sanitization is the mechanical or physical removal, which is going to reduce the number of microorganisms. You want to remember here that dilution is the solution to pollution. And you're going to see as we move forward, especially with gram-negative bacteria, it is important to decrease the number of bacteria oftentimes before we treat. Okay, so we want to reduce the number of microorganisms, also other organic debris. This is going to be done in restaurants and commercial settings for utensils that must be reused. Soap, detergents, and commercial dishwashers are usually used. Hand washing is mechanical removal. Antisepsis or degermation is just a decrease in the numbers of microbes only on living tissues. Hand washing and scrubbing falls in this category as well. Immersing in chemicals like alcohol wipes before an injection or betadine swabs before blood donation, that type of thing. Surgical hand scrubs. It's the use of chemical agents on exposed body surfaces to inhibit those vegetative pathogens. So our primary targets of microbial control are going to be microorganisms capable of causing infection or spoilage in the environment or on the human body. The targeted population often contains mixtures of microbes with extreme differences in resistance and harmfulness. Bacterial endospores, those have been traditionally considered the most resistant microbial entities. So those are going to be at the top. The goal of any sterilization process is the destruction of bacterial endospores. All right. Any process that kills endospores is going to kill everything else since it is the most resistant. Okay. So this is a really good chart. You definitely need to know this. This is the resistance of different microbial types. Starting from the highest in resistance to the lowest. And so you can see that prions and endospores are going to be the hardest to kill. Vegetative and bacteria and fungi are the easiest to kill. So be sure you know this. I can promise you this is on the test. All right, so we're going to go over details of these, but these are some different control agents, and this is going to be comparing the resistance of things like bacterial endospores or those vegetative cells to these control agents. Agents that denature microbial proteins include moist heat, alcohol, acids, metallic ions, but not X-rays. Okay, bacterial, and that's important. Bacterial endospores are traditionally the most resistant, especially compared to vegetative cells. So we're looking here, we've got sterilizing gas, so that's our ethylene oxide, that's going to be sporicidal. Moist heat, sporicidal liquids, but remember, not X-rays. These are not going to, X-rays are not going to be really good enough to hit our endospores very well, okay? So we need to differentiate between sterilization and disinfection, agents versus processes. These are some of the other terms here that describe agents. CIDE, or side, means kill. Okay, bactericide is a chemical that will destroy bacteria that aren't in the endospore stage. So it will kill when you hear side. Okay, and if you know what I mean by that, you know. If you don't, sorry, you're not watching the right TV show. Anyway, so fungicide kills fungal spores, hyphae, and some yeast. A viricide, obviously that's going to kill viruses. Well, actually, I should say inactivate viruses, because you can't kill something that's not living. Sporicide is going to destroy bacterial endospores. An example of that there again is ethylene oxide. It's sporicidal. More terms here, and if I'm going over these terms, that should let you know this is a definition. Definitions you should learn just no matter what. But if I'm going over a definition, then that should tell you you really need to know it, okay? So antiseptics, this is going to be a chemical agent applied directly to an exposed body surface. All right, so skin and mucous membranes. Mucous membranes are anything that opens up to the outside. Wounds, surgical incisions, these areas, in order to prevent vegetative pathogens. Preparing the skin before surgical incisions with iodine compounds is an example. Swabbing an open root canal with hydrogen peroxide. Hand washing with a germicidal soap. Okay, all falls in that category. Sepsis is going to be the growth of microorganisms in the blood and other tissues. Okay. Asepsis is a practice that prevents the entry of infectious agents into sterile tissues and keeps you from getting that infection. And an aseptic technique, that's what you're going to practice in healthcare. That can be sterilization all the way to antisepsis. Stasis, or static, means to stand still. These can be used in combination with prefixes to indicate a state where microbes are prevented from multiplying, but not necessarily killed. All right. So microbistatic doesn't kill but it temporarily stops multiplication. Okay. Even a cidal agent does not necessarily result in sterilization, depending on how it's used. We've got bacteriostatic. That's going to be a chemical agent that stops the growth of bacteria on a tissue or an object. Fungistatic stops or inhibits fungal growth. Okay. Lots of factors are important when you're working with microbial control. We've got to determine how devices come into contact with patients, and we classify these as critical, semi-critical, or non-critical devices. All right. Critical come into contact with sterile tissues, so sterilization must be done before using. Things like a hypodermic needle. This is overcoming or breaking through the first line of defense, all right, which is our skin. Semi-critical comes into contact with mucous membranes. Remember, a mucous membrane is something that lines the opening, something that opens to the outside. So we think nasal passages, but this is also your ears, your vagina, anal, stomach. All of those things are actually mucous membranes, okay? So techniques like endoscopy. That's a portal of entry. We are making a portal of entry that bypasses our first line of defense. So these should have a higher level disinfectant or sterilization. Okay. Non-critical do not touch the patient or bypass the first line of defense. So these only require minimal disinfection. This is going to be things like crutches or blood pressure cuff. So again, let's define some terms here. Microbial death. Death is the permanent termination of an organism's vital processes. Microbes have no conspicuous vital process, therefore death can be a little bit difficult to determine. Microbial death happens when you've got that permanent loss of reproduction. Cells in a culture, for instance, die at a constant rate. Permanent loss of reproductive capability. Even under optimum growth conditions, this has become the accepted microbial definition of death, is permanent loss of reproductive capability. So this is really important. Active cells continue to die more quickly than less metabolically active cells, and eventually a point is going to be reached where the survival of cells is highly unlikely. Okay, so this point is equivalent to sterilization. So what factors can influence how well an antimicrobial agent works? Okay, number of organisms. The higher load of contaminants, the longer it takes to destroy them, right? So the more you have, the longer it's going to take. The nature of the microorganism in the population. The target population is usually mixed bacteria, fungi, spores, and viruses, so it may have a broad spectrum of resistance. Temperature and pH of the environment, all right, very important. That could counteract a particular antimicrobial agent or maybe enhance it. Usually a counteract, okay? The concentration, dose matters, intensity of the agent, all right? Concentration, UV radiation is most effective at 260 nanometers, okay? Most disinfectants are more active at higher concentrations, but more is not always better. Isopropyl alcohol works best at 70%. If you get above 90%, it is 70%, it is actually less effective. So dose matters, all right? Mode or method of action, how does it kill or inhibit the microorganism? Presence of other solvents, organic matter, or other inhibitors, okay? Things like saliva, blood, feces. These can actually inhibit the action of the disinfection. So like organic matter can be in the way. If you can't reach the substance, you can't treat the substance, right? This is why you must clean wounds out. So how do antimicrobial agents do their job? Okay, they have a wide range of targets in the cell, cellular targets. So the least effective agents tend to be, the least selective agents tend to be effective against the widest range of microbes, like heat or radiation. Okay, selective agents target a singular cellular component, like a drug. All right. So what is the method of action? How does the agent work? So we can affect these 4 different areas here, okay? The cell wall, all right? The cytoplasmic membrane, synthesis of DNA or RNA, or synthesis of proteins. Again, so good slide here. The target, the effects of the agents, and some examples used. So we're going to look in more detail at specific things. But the cell wall, agents can damage the cell wall by lysing it or causing it to rupture, digesting it, or stop it from even being made, okay? Chemicals, detergents, and alcohol often do this. Cytoplasmic membrane. You can actually bind to that lipid layer of the cytoplasmic membrane and open it up and allow chemicals to get into the cell or ions to come out of the cell. So you're basically changing the permeability. Detergents are really good at this. Cellular synthesis. You can stop synthesis of proteins, okay, in different ways. You can stop growth and metabolism, and you can also cause a mutation. Things that do this are formaldehyde, radiation, ethylene oxide, okay. Proteins. You can denature proteins, which breaks them apart and renders them non-functional. You can attach to the active site of an enzyme protein and prevent it from interacting with its substrate. Things that do this are going to be moist heat, alcohol, and phenols. So surfactant that can actually affect the cell membrane. They can disrupt the integrity of the membrane by inserting into that lipid bilayer, disrupting it, and creating these channels down here that you can see, allowing things to come in or out of the cell. Okay, this is how soap works. Soap is a surfactant. What happens when you put soap in a sink full of dirty grease? Okay, that grease scatters, right? It disrupts it. That's a surfactant. So another method of physical control here is moist heat and dry heat. These work a little bit more effectively in certain situations. Elevated temperatures, a higher temperature that is more than the maximum growth temperature is going to be microbial-cidal. Cidal, remember, means to kill. Okay, a lower temperature below the minimum growth temperature are microbistatic. Okay, so they're going to inhibit growth, not necessarily kill. Moist and dry heat differ in their mode of action and how efficient they are. Moist heat is going to be a form of hot water, boiling water, or steam about 60 to 135 degrees Celsius, operates at a lower temperature and shorter exposure times to achieve the same effectiveness as dry heat. So often moist heat is better, all right? Microbicidal effect is going to be coagulation, denature of proteins, okay, that type of things. Dry heat is going to be hot air or an open flame. And so you've got a higher degree of heat, 160 degrees Celsius to even several thousand degrees. This is going to dehydrate the cell, remove water that is necessary for metabolic reactions. It can denature proteins. It can increase the stability of some protein conformations, requiring higher temperatures. At very high temperatures, it can oxidize the cell and, of course, burn them completely to ashes. So it is more intense, but it is actually a little less efficient than moist heat. So let's talk a little bit about denaturing, okay? And this is the action of heat and chemicals on proteins. We have what we call the native state of a protein, and that is formed by the bonds that create active sites to fit the substrate. So when a protein is made, all right, it's going to come out and it's going to automatically fold into this globular shape, okay? Well, you can see here that there's a specific shape where this substrate is going to fit and act on. So this is what we call the native state. This is the state that it works, the functional protein. So if it's completely denatured, you can see here, it's totally non-functional. It's like pulling out a slinky and it's never going to go back again, okay? It can also sometimes just cause a change in conformation. So this substrate is no longer going to fit there, okay? The active site no longer will fit the substrate, okay? So that can cause either unfolding or random bonding and incorrect folding. And so that's what this is showing you here. It can have other things bond and cause it to be blocked in that manner. So comparing here endospores versus those vegetative cells. Vegetative cells are just as of endospores are just as susceptible as vegetative cells of non-endospore formers. Okay, so remember we have bacteria that can form endospores. If they're in the vegetative state, they're just as susceptible whether they can form that endospore or not. Okay, other microbes like fungi or protozoa are similar to their sensitivity. Viruses are very heat tolerant. Okay, very heat tolerant. So endospores, however, have the greatest resistance. It usually requires very long-term high temperatures above boiling to kill an endospore. Vegetative cell, those are going to vary in sensitivity. Sometimes you can do 50 degrees Celsius. Now, granted, 100 degrees Celsius is boiling for 3 minutes, or 60 degrees Celsius for 60 minutes. And that's just a variation between different microbes, okay? But remember that those viruses are very heat resistant. Their tolerance extends from 55 degrees Celsius for 2 to 5 minutes to up to 60 degrees Celsius for 600 minutes. So viruses are much harder to kill with heat. All right, these two terms are important. Be sure you know these. Thermal death time is the shortest length of time required to kill all microbes at a specific temperature. Thermal death point is the lowest temperature required to kill all microbes in a sample within 10 minutes. Okay, so similar but not exactly the same. So we've got three methods of moist heat applications. We have boiling water disinfection, which is what is seen here at the top. 100 degrees Celsius doesn't kill all the resistant cells. This is not sterilization, okay, because it doesn't kill all of them. 30 minutes will kill most non-spore forming pathogens. And one of the downfalls is it can be contaminated when removed from the water. Okay, so if you put something in boiling water, you take it out, it can easily get contaminated again. Next, we have pasteurization. That's for liquids like milk, juice, beer. These can be contaminated easily. So heat is used to reduce numbers, right? Dilution is the solution to pollution. This is going to reduce the microbial load and destroy pathogens, okay? Pasteurization is where heat is applied to liquids to kill agents of infection and spoilage. It reduces the number of vegetative forms, okay? Milk is not sterilized after regular pasteurization. This is why when it's unopened, it still can spoil even if it's not opened. So this is showing, sorry, I don't know if that recorded or not. It looked like it paused. This is showing an autoclave, and we have this in the lab, and we use this sometimes. This is steam pressure. You could also have this at home. Very popular of late is the Instapot, which is a pressure cooker, steam and pressure, okay? Higher temperature is pressured for items in this. It is most efficient at 15 psi, which is pounds per square inch for 121 degrees Celsius. So it uses high pressure and temperatures in combination together to deal with a heat resistant material like a surgical dressing, a metallic instrument, that type of thing. So dry heat is not as widely used as moist heat, but it is still important. All right, we have two methods. We have incineration, and this is what we do with our Bunsen burner, which you're doing in your simulations, and we do in the lab. The temperatures reach up to 1,870 degrees Celsius at the hottest portion of the flame, which is the tip of the going to reduce a microbe to ash. It is fast and effective, but the limiting factor is it's got to be done with metal or heat-resistant glass material. So it's pretty limiting there, okay? Then we also have a dry heat oven. This is going to be circulated air. It's going to transfer heat to the materials in the oven. Sterilization is going to require 150 to 180 degrees Celsius for 2 to 4 hours, okay? This does destroy endospores. Well, we've talked about heat, so what about the cold? Well, cold can only be counted on to be microbiostatic, not microbicidal. Sorry. So it's not going to kill them, it's just going to slow them down. But the benefit of cold treatment is to slow the growth of cultures and microbes in food during storage. Really important, okay? So it's going to slow down those metabolic processes. And of course, it depends on the microbe. Temperatures range from -70 to -135. And these can actually preserve cultures of bacteria. We do this, we put them in the fridge to slow them down. Virus, fungi for long periods of time. Two more methods here, desiccation and lyophilization. Desiccation means drying out. This is where vegetative cells are going to be exposed to normal room temperature and become dehydrated. This happens a lot. Every year at the beginning of the year in micro, I get new bacteria samples. I put them out on plates and I grow them and I put them in the drawer. By the end of the semester, and definitely by the end of the year, they have dried out completely. Some microbes are killed by desiccation, but not all. Some sometimes are even preserved, okay? Lyophilization is going to be freeze drying, all right? So this is a method of preserving microorganisms in a viable state for a very long time. A pure culture can be frozen very quickly, instantaneously, and then exposed to a vacuum that's going to suck the water and air out, okay? And so there's no ice crystals that form, and that pretty much will help you to store them indefinitely. We can also kill with light, radiation. This is energy emitted from atomic activities and dispersed at a very high velocity through matter or space. This is a very useful source of antimicrobial activity. I have had a goggle case that you can turn on a UV light for 15 minutes. I think it's 15 minutes, and it will kill all germs. Okay, hospitals use specialized UV light in order to disinfect. Sorry, I couldn't think of the word. We have ionizing radiation. This is going to be very effective for sterilization. That's the word I was looking for items that can't withstand heat or chemicals. Those plastic goggles, we can't put heat or chemicals on them or on those. Okay. And so we have gamma rays, X-rays, and UV light. All right. Gamma rays, well, foods are going to undergo irradiation to kill pathogens, insects, and even worms without harming the food or making it radioactive, okay, or slightly cooking the foods. That's pretty handy. UV light can kill fungal cells, vegetative cells, spores, viruses, and even protozoa. Another method to remove microbes is filtration, and we use this to prepare liquids that cannot undergo the heat. Things like blood serum, blood products, vaccines, drugs, IV fluids, enzymes, and other media. Okay, so this is an alternative method for decontaminating things like milk and beer without changing their flavor. This is really important for things like water purification. You're unable to remove soluble materials or toxins that can cause disease. So it's only partially beneficial for certain things. Things like high efficiency particulate air filters, that's a HEPA filter. These are used in hospital rooms and for sterile rooms, okay? And so that's where filtration is going to come through, or this is where it's going to be applied. Lastly, we can use osmotic pressure, and this is where you use a large amount of things like salt. This is one of the old-fashioned ways to cause plasmolysis. You can add a lot of salt or a lot of sugar. What do they do to cure ham? Well, they put a lot of salt or a lot of sugar on it. Think back to when you were little, possibly. When I was little, we would find slugs. I grew up in Louisiana, and they were everywhere. We would pour salt on them. What happened? Well, water follows salt. Water would come out of the cells and it would kill the particular slug, or it can kill bacteria. So we've been using this for centuries to preserve foods. This is not, however, sterile.